Welcome back. In this chapter 8, we're going to talk about pathogenic microorganisms. So what are some of the different types of pathogenic microbes? Well, there's actually 5 major classes that we'll discuss here in this chapter. One are the viruses, of which of course we've heard lots about just recently. Secondly, we'll talk about the protozoan parasites. This is one of my favorite protozoans. Looks like he's got a little monkey face, and this is Giardia lambdali. This represents our helminths. These are some roundworms, and our helminths include the worms and the flukes. Here we have bacteria. Here's the fungus. And I put this picture in simply to show that here are some intracellular organisms. We usually think about the viruses being the only intracellular organisms that replicate inside the cell, but these actually are going to be Rickettsia rickettsii, responsible for Rocky Mountain spotted fever, that are replicating inside of these host cells. So let's look at our bacteria first. How do we classify our bacteria? Well, they are classified based upon 4 major characteristics. First of all, we're going to look at shape and arrangement. We have 3 primary shapes within the bacteria. The coccus, which are spherical shaped bacteria, bacillus, which are the rod-shaped bacteria, and the spiral organisms, of which we have 3 subgroups. The Vibrio, which kind of are comma-shaped, the Spirillum, which have about 3 to 5 twists along their long axis, and the spirochetes, which are really twisted. Of the arrangements, we can see Staphylo, which means they're in grape-like clusters, Strepto, which means they're in chains, and Diplo, which means we have 2. Of the Staphylo, we only will see Staphylococcus. The bacillus do not form these groups. Strepto, we can see Streptococcus or Streptobacillus. Di means 2, so Diplococcus or Diplobacillus. Secondly, we will look at their Gram-stain reaction. Are they gram-positive organisms or are they gram-negative? Those of you that have had micro are familiar with these. Then we can look at their biochemical and growth characteristics. This will include things such as, are they aerobic or anaerobic respirators? Do they form endospores or not? And what type of foodstuff do they need in order to form their energy and proteins? And then lastly, we can look at the antigenic structures. And these are antigens that are found in the cell body, the capsules that some bacteria can form, or the flagella. Such as with E. coli 0157H7. The 0157 is a particular antigenic protein found in the cell wall. The H7 is actually a specific flagellar protein found on that particular organism. So let's look at some of these. So here we have a picture of some bacterium. And so what is the shape and arrangement? So let's look at the shape first. So these typically are all round, and so they would be what? That would be the coccus. The arrangement, for the most part, they're grouped in clusters. And so this would be a staphylococcus. What is the shape of this particular bacterium? And so here's one single bacterium. And you can see that they have about 3 twists along the long axis of the organism. And so this shape would be a spirillum, one of our spiral organisms. What's the shape and arrangement of this particular bacterium? This is a fairly low power view, but you can see that they pretty much are longer than they are wide, and so this shape would be bacillus. The arrangement, they're typically seen in these strings or chains, and so this would be a streptobacillus. Now, this one is a little bit different. I'm going to tell you that all of these organisms are the exact same organism. So, what shapes do we have? Well, we have some that are round or coccus in shape. We have some that are elongated or rod-shaped, so bacillus. And then we have these interesting little fellows. And no comments about the particular shape here. So some bacteria don't really know what they want to be when they grow up. And we call these organisms polymorphic. So let me get my pen up here for just a second. So what does polymorphic mean? Well, pleo means many. Morpho means body. So polymorphic literally means that they have different shapes. And so this is a polymorphic organism, and this would we'd actually refer to as a coccobacillus because we see both coccus and bacillus. So what's the shape of this bacterium? Well, again, we can see an individual right here, and we have many, many twists along the long axis. So again, this is going to be one of our spiral organisms, and this is actually a spirochete. What is the shape and arrangement of this bacterium? So again, we see the coccus, and we see strings such as a pearl or a strand of pearls, and so this would be a streptococcus. This one I put in to show you again another polymorphic organism. This one again has kind of a coccus shape with a long tail on it, and so this would be a coccobacillus. This is actually one of the Clostridium bacteria that forms kind of this tennis racket shape. Now, the shape of this organism is actually a little bit comma shaped. So it has basically about a half twist along the long axis. And this is actually one of our spiral organisms called Vibrio. So in the spiral organisms, how twisted are you? If you have basically just kind of a half twist and you form the comma shapes like these guys, you're a Vibrio. If you have 3 to 5 twists along the long axis, then you're going to be a spirillum. If you have many, many twists, then you're a spirochete. Bacillus organisms actually can be classified as whether or not they have square ends, such as with Bacillus anthracis, rounded ends, which we would see in Mycobacterium tuberculosis organisms, club-shaped, which would be a Corynebacterium, where they're kind of a little elongated at the end and a little bit more large, and fusiform bacteria, which basically take on kind of an odd shape with the bacillus, and the fusiform bacterium species actually are found in this class. Now, in this slide, I've listed our spiral organisms. So again, the comma shape is that Vibrio, where you have kind of that half comma shape. The tightly coiled would be the uh, Spirillum, and a good example of this is Treponema pallidum, which is the causative agent of syphilis. And then a relaxed coil, a good example of this would be Borrelia burgdorferi, which is the causative agent of Lyme disease. And I didn't list here then a Spirillum, which again is that 3 to 5 twists. So what is gram staining? Gram staining is one of the major classifications of our bacteria, and bacteria are either classified as gram positives or gram negatives based on the ability to retain or resist certain dyes. This is based on the chemical and physical properties of their cell walls. We have two stains that we use. The primary stain is called crystal violet, and as the name says, it allows the cells to become violet. Once we have fixed our bacteria on our slide in some method, and usually with gram staining we use heat as a fixation method, we then apply crystal violet. Everybody will stain purple at this point because everybody takes up the crystal violet. We then will add iodine. Iodine is what we refer to as a mordant. A mordant highlights the primary stain. Iodine, even though it does have a color to it, will not add any color. And so still everybody will be purple at the end of the iodine step. The critical step of gram staining is the decolorizing step. We use a 50% acid alcohol solution, and what we'll see is that some of these bacteria will retain the crystal violet while others actually will lose the crystal violet. Then we are going to counterstain with the secondary stain called safranin. It's a reddish stain, and so those cells that decolorized in the third step will take up the safranin stain and appear reddish. Those that retain the crystal violet will remain purple. Those that retain crystal violet are gram positives, and I always remembered it this way: positives are purple. Those that lose the crystal violet in the decolorizing step and take up the safranin and display it will be known as gram negatives. And so the red color has kind of a negative connotation, and that's the way I remembered those. So we dry a fixed suspension of bacteria and put it on a microscope slide and fix it by heating it. Crystal violet, our primary basic stain, will be added, and it will sit for a certain amount of time and then be actually washed off with using deionized water. We then will add the grams iodine that acts as the mordant. Again, everybody will be staining purple after this step. This third step, however, the decolorizing step, is the important one. And again, usually we use about a 50% alcohol with an acid, and most of the time we use acetone as this acid, and this causes rapid decolorization of the gram-negative bacteria. Then we add the secondary safranin stain, which is an acidic red stain, and those gram negatives then take up the safranin stain. So gram positives again resist decolorization and retain the crystal violet and remain purple. The gram negatives decolorize and stain red with the safranin. So what would be the gram reaction of these organisms? First of all, again, remind yourself what is the shape and arrangement. So these are Staphylococcus, and they all look pretty purple to me, so these would be a gram-positive Staphylococcus. Now, we're looking at the organisms within these neutrophils here, and the neutrophils are staining a fairly reddish color, and these organisms, actually, though they look purple, actually are staining a reddish color as well. And so these are actually a gram-negative Diplococcus. And in fact, this is Neisseria gonorrhoeae found within the neutrophils. So what would be our gram reaction here of these bacillus? Well, most of them are staining fairly purple, and so this is a gram-negative bacillus. Versus this one, which is certainly reddish in color, And so these are a gram-negative bacillus. So let's take a look at some of these common organisms. So if we're looking at gram-positive coccus, we have primarily three genuses: the Staphylococcus, the Streptococcus, and the Pneumococcus. Of the gram-negative cocci, we have the Gynococci and the Meningococci. Both of these are within the Neisseria genus, so we have Neisseria gonorrhoeae and Neisseria meningitis. Of the gram-positive bacillus that cause disease, Corynebacteria, which causes diphtheria, Listeria, which causes listeriosis, the bacilli bacteria, of which there are a number of these, and the Clostridia bacteria, which again, there's a number of these. The bacilli and the Clostridia are both what we call endospore formers as well. Of the gram-negative bacillus, you can see that there's a huge list. Haemophilus pneumoniae is one of our major pneumonia bacteria. Gardnerella, Francisella tularemia causes tularemia. Yersinia pestis. This is a historically important bacteria because this is the causative agent of bubonic plague. Brucellosis, Legionella, Salmonella, Shigella, Campylobacter, the cholera bacillus, which I'll talk about here in just a second again, and then colonic bacteria. And this includes E. coli, but can also include Salmonella and Shigella. Of the spiral organisms, a couple of important ones that are gram-positive in the pathogenic bacteria are Treponema pallidum, that causes syphilis, and Borrelia burgdorferi, that causes Lyme disease. Now again, I put this guy, cholera, down here in the spiral organisms because the causative agent of cholera is Vibrio cholerae. So I would actually put this one down here in the spiral organisms. And then we have a group of organisms called the acid-fast organisms. These will be gram-positive on the gram test, but when we use a stain called the acid-fast stain, what we actually will find is that Mycobacterium tuberculosis and Mycobacterium leprae actually will stain with the acid-fast staining process. They will stain a reddish color. This is because Mycobacterium actually has another protein in their cell wall called mycolic acid, and this causes them to be acid-fast. So let's look at some of these organisms and the diseases they cause. Staphylococcus aureus. This causes various localized and systemic infections and is one of the more common skin diseases that we see in humans. The Streptococcus, we have two groups: the beta hemolytic Streptococcus, group A, which is the primary group that causes streptococcal infections in the throat, so pharyngitis, and the beta hemolytic Streptococcus group B that are primarily seen with urinary tract and wound infections and systemic infections and infections in newborns. And then other Streptococcus, which are not included in these two groups, that can also cause wound and urinary tract infection. Streptococcus pneumoniae has actually been reclassified as a pneumococcus, and it's very commonly seen in middle ear and sinus infections, other systemic infections, and one of the common causes of lobar pneumonia in the lungs, as well as potentially causing meningitis. Neisseria gonorrhoeae causes gonorrhea. Neisseria meningitis causes meningococcus meningitis. And these are actually the gram-positives right here, and the Neisseria are the gram-negatives. Of the aerobic gram-positive rods, Corynebacterium diphtheriae, which is the clinical etiological agents of diphtheria, and Listeria monocytogenes, which causes listeriosis. And both of these are fairly rare, but we do see both of them occasionally. And then of the aerobic spore-forming rods, we have Bacillus anthracis, which is one of our major pathogens. And this is primarily disease of animals, but people can become infected from contact with the wool or animal products. There are three forms of anthrax in humans. The most common form is the cutaneous form, which fortunately is the most curable. The deadly form is the inhalation form of anthrax, which is the one that we heard about in the early 2000s with the anthrax spores going through the mail. And a GI form of anthrax, which is fairly rare in humans. So this picture actually shows some skin lesions, and if we culture these, this would actually be Staphylococcus aureus. This is a nice picture in the back of the throat, and this is what would be commonly seen in strep throat. Here's the tonsils, that's the uvula, and you can see the inflammatory process that's going on there. Neisseria gonorrhoeae is actually an intracellular organism, so the little tiny red spots here in the neutrophils are the organisms. It actually exists as a diplococcus. It's just a little hard to see on this one, but you've got one pair right there and a pair there. So this is what the organism actually looks like. This is a picture of actually diphtheria. Diphtheria is not seen much anymore simply because we do have our diphtheria vaccine. But one of the things that we'll see commonly with a Corynebacterium diphtheria infection is the formation of something called a pseudomembrane in the back of the throat. And this is really just a very thick mucus plug that forms back there. And then this, seen on the surface of the skin, is classic cutaneous anthrax. Now, there is an error on this chart. These are not cocci, these are actually bacilli. And these are the anaerobic spore-forming rods. And this is the Clostridial group. There are four major Clostridium species that cause disease in humans and animals. Clostridium perfringens causes gas gangrene. This actually will cause muscle necrosis and hemolytic anemia. And gas formation is in infected tissues, and I'll show you a picture in just a moment. Clostridium tetani is the etiological agent of tetanus, or the common name lockjaw. Clostridium botulinum causes botulism, one of our forms of food poisoning. And Clostridium difficile, or C. diff, as it's commonly referred to, causes usually an antibiotic-associated colitis. All of these clostridial organisms produce toxins that actually cause the tissue damage. So if you'll see here, toxin, 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 and this C. diff actually produce toxins that oftentimes will cause bowel damage in the bowel mucosa. Of the gram-negative bacillus that cause disease, Haemophilus influenzae I mentioned causes pulmonary infection, but also Haemophilus influenza is one of the major bacterial causes of meningitis in children. Yersinia pestis causes bubonic plague. This organism is actually transmitted to humans by fleas. And during the Middle Ages in Europe, when we had the bubonic plague outbreak, there were millions of people that died, and they thought at first that the wild rodents or rats were transmitting this disease to people. And so they literally burned down whole cities. The problem was when they killed the rats, the fleas jumped on the closest blood donor, which happened to be the human, and this actually then caused this disease to spread even more quickly. Can spread from person to person if the infection spreads to the lungs, which is a form called pneumonic plague. The brucellosis species, of which there are several different types, causes brucellosis. This is a febrile illness that is usually from contact with infected animals or the consumption of raw milk or other raw products from infected animals. And then Francisella tularemia causes a disease known as tularemia. This is a febrile illness and usually is again acquired from ingesting the flesh of infected animals, or usually this is wild rabbits, or can actually has been shown to be transmitted to humans by the bite of infected ticks or deer flies. So this is a picture of gas gangrene, and you'll notice that there's a lot of inflammation going on here in the thigh. But when we take a look at the radiograph, there's the femur, and you'll notice that there's a lot of black within the tissues. That black is actually trapped gas that's produced by Clostridium perfringens. And if you were to run your finger down the surface of the skin, this would feel almost like tissue paper crinkling, and we call that process crepitus. This picture you may not recognize real well, but certainly if you look especially at the facial expression right here, this is classic positioning of Clostridium tetani or tetanus. The upper limbs usually will go into tectonic contractions and flexions, simply because our flexor muscles are stronger than our extensors, and we typically will see hyperextension of the neck. This is what tetanus looks like in a dog, and you'll notice that there's full extension in all four limbs. There's that hyperflexion of the neck, or sorry, hyper extension of the neck. This is actually pneumonia or Haemophilus pneumoniae, and this is just the classic pneumonia pattern that we see in the lungs. It looks very cloudy, and this is due to fluid within the lungs. This is actually a picture of a gentleman from Oregon that died of this particular disease. And this is Yersinia pestis. You'll notice that there are swellings here. Those are lymph nodes that have swollen. This one is actually ruptured to the outside of the body. And these are what's known as buboes. This is what gives this name the bubonic plague. And this is actually going to be a Francisella. This is a common lesion that we see with tularemia in humans. This is actually the liver from a wild rabbit, and you'll notice that there's these little spots within the liver, and those little spots are actually colonies of Francisella tularemia. Some more of these bacillus, and again, this is an error here, so these are actually gram-negative bacilli. Legionella pneumophilia causes Legionnaire's disease, which is a respiratory disease. Salmonella, of which there are several different species, causes primarily gastroenteritis. Typically, salmonella is acquired from contaminated food or water sources. So we typically say that salmonella is transmitted by a fecal-oral route. Shigella species, same thing as salmonella, usually contracted by contaminated food or water, can however be spread by person-to-person contact. The Campylobacter species again cause gastroenteritis, may cause systemic infection. Usually we get this from, again, contacting contaminated water or eating undercooked meat or poultry or contact with infected animals. And then kind of the big poster child of these four big enteric bacteria, we call them, is E. coli. Gastroenteritis, but can also cause urinary tract infections and systemic infections. Some strains, and the big one is E. coli 0157H7, produce toxins that can cause something called hemolytic anemia and kidney damage, which is known as hemolytic uremic syndrome, or HUS. In the spiral organisms, Treponema pallidum causing syphilis, one of our sexually transmitted diseases, and Borrelia burgdorferi transmitting Lyme disease, and this is transmitted by ticks, and primarily the ticks species is Ixodes genus, and Ixodes scapularis is the tick that we have here in southwest Missouri that can transmit Borrelia burgdorferi. Of the acid-fast bacteria, Mycobacterium tuberculosis causing pulmonary tuberculosis, Mycobacterium avium complex, which is usually a disease seen in birds, the avium, but can be a opportunistic infection in AIDS patients. And I'll put one more on here of the acid-fast mycobacterium, and that is Mycobacterium leprae, causing leprosy. This is a classic pattern of the respiratory disease in Legionnaire's disease. This disease actually is a respiratory-borne illness and was actually named for the Legionnaires that got it at a convention up in Pennsylvania in 1976. Our enteric bacteria, E. coli, Salmonella, Shigella, Campylobacter, all of these again primarily cause GI upset. This is actually primary syphilis, and this erosive lesion right here is known as a canker, and it's the common symptom that we see in primary syphilis caused by Treponema pallidum. Here's Borrelia burgdorferi, one of our spirochetes, and this is the classic what we call target lesion. The tick bite would have been right in the center of this. However, some Lyme disease patients never present with this target lesion, but simply have a rash. These are lungs that have tuberculosis granulomas. Tuberculosis causes a chronic inflammation, and so these areas right in here are actually granulomas, and we'll see tuberculosis actually growing inside macrophages within these granulomatous lesions. So let's look a little closer at some staphylococcal diseases. Staphylococcus is actually normal inhabitants of the skin. So primarily on our surface of our skin, we have Staphylococcus epidermidis. Can be seen in the nasal cavity, and this can include Staphylococcus aureus, and also Staphylococcus aureus can be on the skin as well in low numbers. So commonly found on skin and in the nose of patients and hospital staff. Now, you've probably heard a lot about methicillin-resistant Staphylococcus aureus, or MRSA. At this point, probably most hospital staff have MRSA in their nose, and many patients probably do as well. Normally, Staphylococcus epidermidis is not pathogenic. Staphylococcus aureus is a pathogenic staphylococcus, but typically becomes opportunistic when given the opportunity to get through the surface of the skin or mucosal membrane. Diseases that Staphylococcus causes is usually caused by toxins that the bacteria produces. Some Staphylococcus may result in vomiting and diarrhea within the GI tract. Toxic shock. This was a disorder that was historically related to the use of tampons in females, but toxic shock syndrome can actually be caused by just a systemic Staphylococcus infection. And in toxic shock, we can see headache, fevers, and these can get actually very high, up to 105 degrees. So we see sometimes confusions, sore throat, a non-purulent conjunctivitis in the eyes, lethargy, vomiting, and interestingly enough, this can cause a sunburn-like rash, and what we'll actually see sometimes is actually sloughing of the skin, especially on the palms of the hands and the soles of the feet. This can actually come on very rapidly and have rapid progression, and in fact, we can actually see a hypotension, syncope, and shock, and death occurring very quickly in toxic shock. Staphylococcus can result in tissue necrosis. It can cause hemolysis of the red blood cells. And what we'll see is that we get inflammation with these Staphylococcus infections. So skin infections, impetigo, boils, and this can be furnicles or carbuncles, and this is just simply small lesions that have staph versus larger lesions. Nail infections called perionychia. Cellulitis, where the Staphylococcus actually spreads through the subcutaneous regions and fascial planes between muscles. Staphylococcus is a common surgical wound infection problem, can cause eye infections and mastitis in females that are lactating or sometimes not even lactating. Can cause extreme sepsis throughout the bodies from wounds or IV drug use. Can result in endocarditis, causing an inflammation in the lining of the heart and the valves. And this can be because of normal or prostatic valves that have been put in, or IV drug use where the Staphylococcus gets direct access through the bloodstream. Can result in osteomyelitis and arthritis, so infection in the bone and joints. Pneumonias can result in abscess formation. And again, some strains are highly resistant to most of our antibiotics, and this is the methicillin-resistant Staph aureus, or what we now refer to MRSA is multi-resistant Staph aureus, because most of these MRSAs, simply no antibiotic is going to work against them. Streptococcal infections. Streptococcal infections we base on type of hemolysis of blood and the differences in the carbohydrate antigens in their cell walls, or what we call the C carbohydrates. And we have a group called the Lancefield classification of groups A to U. We primarily talk about group A and B. Beta hemolysis is simply complete lysis of red blood cells, either in the blood or in a blood agar plate. The group A, beta hemolytic streps, which is primarily including Streptococcus pyogenes. This is the group that causes, again, strep throat or pharyngitis. Whereas group B, Streptococcus agalactia, causes streptococcal infections in the genital region, causes neonatal meningitis, and can even result in sepsis throughout the body. Now, I will mention group D because we do now have this organism reclassified, Enterococcus faecalis. And as the name says, faecalis, this guy causes problems in the GI tract. Streptococcus bovis, which is originating from cattle, and both of these can result in urinary tract infections, biliary infections in the gallbladder, or cardiovascular infections. Streptococcus is a normal inhabitant of the skin, mouth, and pharynx. It's also found in the gut and in the female genital tract. Most of these normal inhabitants, such as Streptococcus viridans or the Peptostreptococci group, are non-pathogenic but may become opportunistic given the opportunity. Some diseases that we see is pyogenic pharyngitis, again, strep throat. Streptococcus can cause cellulitis, just like staph. endocarditis, urinary tract infections. Toxigenic, we see scarlet fever, and this is again where we have a type 2 hypersensitivity reaction where the antibodies made to the streptococcal antigen attack similar antigens on the lining of the heart. And streptococcus can also cause toxic shock syndrome. Rheumatic fever, again, also with scarlet fever, the same type thing, and glomerulonephritis. Now, how do antibiotics work? Well, what we see is antibiotics work in one of 4 areas, and I'm going to make this really simple. We either work at the cell wall, inhibiting the either production of the cell wall, basically peptidoglycan in the cell wall, or we break it down. And so some of those that actually block synthesis and repair include the penicillins, the cephalosporins, the carpenamines, vancomycin, bacitracin, and a couple of others. The second area where antibiotics can work is at the cell membrane. And this would include the polymyxins and daptomycin. And what both of these groups of antibiotics do is they cause loss of selective permeability. So now things that normally would be screened out can get in and cause problems in the cell, or things that normally are held in the cell will actually leak out. Third thing is antibiotics can disrupt protein synthesis. They can do this in one of 2 ways. One, they could actually inhibit the replication and transcription process. The fluoroquinolones actually inhibit DNA gyrase that unwinds the supercoiling ahead of the replication fork, and therefore we will not copy or replicate DNA. Rifampin actually inhibits the RNA polymerase, so we cannot make the messenger RNA strand. Others will actually block the ribosome function. Some will block the action of the large 50S subunit, and this includes erythromycin and clindamycin. Some will block the action of the 30S subunit in the ribosome. This includes the aminoglycosides, which includes gentamicin and streptomycin, and the tetracyclines. Some would actually block the entire function of the ribosome, and this includes an antibiotic called linezolid, which is actually used in cases of tuberculosis and actually in HIV treatment. And the fourth thing that antibiotics can disrupt is metabolism within the bacterial cell. And one of the large bacterial pathways that we block is folic acid synthesis. No folic acid, these cells cannot live. And so the sulfonamides or the sulfa drugs, and trimethoprim is often added to some of the sulfa drugs. These will block and inhibit metabolism of folic acid. So antibiotics will affect one of 4 areas: cell wall, cell membrane, DNA RNA synthesis copying along with protein synthesis, and metabolic pathways. Now, how would I know which antibiotic to use? Well, we can do a number of different things, and one of these is called the antibiotic sensitivity test. One of these will be the tube dilution test, and all this does is it measures the highest dilution or the smallest amount of antibiotic that will inhibit the growth in a test tube. And so what we'll do is, again, we will actually then form dilutions of the drug. And so what is the smallest amount or the highest dilution that we can use? So in this particular picture, we see that we have 100 micrograms per milliliter in this tube. We have 50, 25, 12.5. So we're diluting by half every time. So we have a 1 to 2, 1 to 4, 1 to 8, 1 to 16 dilution of the drug. We'll see no growth at 100 milligrams per milliliter, 50, 25 no growth, and at 12.5 no growth. But at 6.25 milligrams per mils, we actually begin to see growth of the microorganism. So we say that the minimum inhibitory concentration, or MIC, of this antibiotic is 12.5 milligrams per milliliter. We can do mathematics on that and actually figure out then the dosage, which would be milligrams per kilogram body weight. We also will see the DISC methods, and what we're looking for in the DISC methods is inhibition of growth around a disc that is impregnated with a particular antibiotic. We have two forms of this, the Kirby-Bauer plate and the E-test. The Kirby-Bauer plate, and I have a video that I've posted under the narrated PowerPoints for you to watch on this. And this is a snapshot from that video. What we'll do is we make a lawn of bacteria on a large plate using an auger known as Mittenhuler auger. We then drop these discs impregnated with these different antibiotics onto the plate and incubate. What we're looking for is the zone of inhibition of growth. So around the gentamicin disc, you can see that no bacteria are growing right there. This means that as the gentamicin leaches out into the media, it will inhibit the growth of these organisms. The trimethoprim sulfa drug here has a fairly large zone of inhibition. But I wouldn't want to use bacitracin or penicillin or vancomycin because there is no zone of inhibition, or erythromycin either. That means that this drug was completely ineffective against this particular bacterium, so therefore this bacterium has a resistance to these drugs. Now, we don't just go, okay, well, I can choose any of these then that have a zone of inhibition. But we can't just look at the zone of inhibition itself. We actually have to look at how big the zone actually is. So here we have an example of a zone of inhibition, and we're going to measure the diameter of that zone. So here's tetracycline. The little 30 means that there were 30 micrograms embedded into the disc. If we measure this zone of inhibition, we're at about 36 millimeters. So if we go down to this standardized Kirby-Bauer chart and go to tetracycline, we see that any zone of inhibition greater than 19 would mean that it is susceptible to this tetracycline. So in this case, this particular bacteria would be susceptible to tetracycline. Here we have some example results from a Kirby-Bauer plate for Staphylococcus aureus. And tetracycline, we measured the zone of inhibition at 25 millimeters, greater than 19, means it's susceptible. But let's look at penicillin G. We only measured 10 millimeters on that zone. We did have inhibition, but it was only 10 millimeters. Greater than 29 would mean that the bacteria would be susceptible to penicillin G, but less than 20 means that it's resistant to penicillin. So 10 is less than 20, so it's a resistant organism. But let's look at erythromycin. We measured the zone of inhibition at 15 millimeters. Less than 13, it would be resistant to erythromycin. Greater than 18, it would be susceptible. 15 is smack in the middle between these two. So we say that this would be an intermediate drug. So what drugs would I not want to use for this Staphylococcus aureus? Well, certainly neomycin, penicillin G, streptomycin, polymyxin B. All of those are resistant, even though they had a zone of inhibition. I would want to choose either bacitracin, chloramphenicol, gentamicin, kanamycin, tetracycline, or vancomycin. I might even decide to choose erythromycin. But as I look at the zones of inhibition, probably I would choose tetracycline simply because it has the largest zone of inhibition. An alternative to the Kirby-Bauer plate is called the E-test or the echelon test. This basically kind of does two things. One, we will see a zone of inhibition if we have any inhibition to these drugs. But the other thing is these strips are actually impregnated with increasing amounts of the antibiotic. So down here we have 0.016 micrograms, 0.23, 0.032, etc. So right up here we're at 1 microgram, and up here we're 256 micrograms. So we'll see that we do have a zone of inhibition, and it kind of forms a teardrop shape, because what this E-test strip will also show us is basically that minimum inhibitory concentration that we saw in the test tubes. So we're about at 1 microgram. We can do the math and actually figure out dosage then based upon this. Now, what are some adverse effects of antibiotics, or are there any adverse effects of antibiotics? Well, there can be. One is these are toxic. Now, if we can choose antibiotics that work on unique things to the bacteria, they tend not to be toxic to us. So things like penicillin, cephalosporins that work to break down the peptidoglycan in the cell wall of bacteria, our cells don't have that, and so therefore they're not going to be toxic to our cells. But others work in decreasing metabolic pathways, or stopping metabolic pathways, or stopping translation, transcription, and protein synthesis and DNA replication, and those could affect our own cells. Certainly hypersensitivity reactions. There are a number of people that can develop hypersensitivity reactions to certain drugs. Penicillin, cephalosporins, erythromycin, tetracyclines all tend to have some severe hypersensitivities in certain patients. We can see alteration of the normal microbiome. This is one of the ways that we get C. diff in the digestive tract. We use broad spectrum antibiotics, and this kills off all the good bacteria, and what little C. diff bacteria we have then will overgrow because the other bacteria have been taken out. We can also see development of resistant strains, and have we not seen this? This could be due to simply spontaneous mutation within the bacteria that results in resistance, or plasmid-acquired resistance through conjugation, the exchange of genetic material from one organism to another. Some bacteria also figure out ways to circumvent the effects of antibiotics. Some develop enzymes that will break down the antibiotics. And many of the gram-positive bacteria have developed the ability to produce an enzyme called penicillinase, or the other name is beta-lactamase. And beta-lactamase actually breaks the beta-lactam ring in penicillin's and cephalosporin structure, and this renders them ineffective. Some can change the structure of the cell wall or the cell membrane, thereby rendering the antibiotic ineffective. Or some can change or find alternative pathways for the internal metabolic machinery. Now, I want to talk about some specific bacteria, and this is the chlamydia bacteria. These are gram-negative, non-motile cocci. They form inclusion bodies in infected cells, and this is actually a cell that is ruptured, and all of these little green cocci structures are chlamydia trachomatis, and this is the causative agent of the sexually transmitted disease chlamydia. Chlamydia are unique among the bacteria in that they are obligate intracellular parasites. They have to reproduce inside a living host cell. And in fact, chlamydia was actually classified as a virus for many years simply because they have to reproduce inside a host cell. They have a rigid cell wall and reproduce by this distinct intracellular cycle in their host cells. They are susceptible to tetracycline and most of the erythromycins. However, we are starting to see a lot of resistance, and there is no vaccine currently available for chlamydia. Chlamydia cause a number of different diseases, one of which is called psittacosis, and this is a pneumonia caused by certain chlamydial bacteria, and this is actually passed in the feces of birds, and so inhalation of dried bird feces. Sometimes we see this in people that have a number of birds within their house. Trachoma, which is actually a chronic conjunctivitis in the conjunctiva of the eyes, it can be caused by chlamydia trachomitis A, B, and C. This can eventually even result in blindness. Non-gonococcal urethritis in men. Chlamydia and gonorrhea cause similar signs in males, causing this urethritis, and so can be spread to other areas, including up through the reproductive tract. In females, chlamydia trachomitis causes cervicitis primarily. This can lead, however, to salpingitis, inflammation of the oviducts or fallopian tubes, can lead to pelvic inflammatory disease, and in fact, PID, one of the major causes of this is chlamydial infections. This could lead to infertility, ectopic pregnancy. And in the neonate, the newborn, we oftentimes will see neonatal inclusion conjunctivitis. The newborn is infected as it passes through the vaginal canal, and this can cause trachomas actually in the newborns, and this can cause severe scarring of the cornea and blindness. And then lymphogranuloma venereum, the venereal disease. This is actually the old name for the sexually transmitted form of chlamydia trachomitis. So here we have some chlamydia that are forming inclusion bodies in these epithelial cells right here. This is the common cervicitis, so this is the cervix here looking through the vaginal canal. Oftentimes you'll see some frothy discharge from the vagina. In males, again, a a urethritis, so we get discharge from the urethra. And this is the inflammation of the labia majora and minora in a female. This is sometimes referred to as genital elephantitis, but this is due to a chlamydial infection. And in males, a lot of times we will actually see a lymphadenitis. So the inguinal lymph nodes in this region will swell due to the chlamydial infection. Now, rickettsia and ehrlichia, these are small bacteria that can cause damage to small blood vessels in the skin, and this results in leakage of blood into the surrounding tissue, causing rashes and edema. And these are what we call the spotted diseases. So Rocky Mountain spotted fever, which is transmitted by ticks. In the East Coast, we see Rocky Mountain spotted fever in the spring and early summer. Typically, these folks will have kind of a flu-like symptom before the rash develops. The rash will then develop usually about 2 to 6 days after onset of the symptoms. Usually begins in the hands and feet and then goes to the trunk. Eventually could even infect the central nervous system. Rickettsial pox, caused by one of the rickettsias, is actually transmitted by a group of mites. Typhus, caused by rickettsia, again, flu-like symptoms with rash, and the epidemic form of this is transmitted by lice. The endemic form of typhus is transmitted by fleas, and the scrub form of typhus is actually transmitted by, again, certain mites. Q fever, one of the rickettsial diseases, is actually an aerosolized transmission, and this causes a pneumonia hepatitis combination. The rash is actually fairly rare, and fortunately, overall, Q fever is actually fairly rare in this country. Ehrlichiosis causes similar flu-like symptoms with usually rashes, and both rickettsia and Ehrlichia are susceptible to tetracyclines or an older antibiotic known as chloramphenicol. However, we do not use chloramphenicol in humans because in 1 out of every 30 to 40,000 people, chloramphenicol will kill the bone marrow, resulting in a non-regenerative aplastic anemia. Rickettsia and Ehrlichia, like chlamydia, are obligate intracellular parasites. And these are transmitted by insects or arachnids, the ticks and mites, to humans. So most of them are transmitted by the bite of an arthropod vector, either ticks, mice, lice, or fleas, except, again, for Q fever, one of the rickettsial diseases. Rickettsia tend to multiply in the endothelial cells of the blood vessels. That's why we get the damage to the blood vessel walls. The Ehrlichia tend to multiply in neutrophils and monocytes. Both causes of febrile disease with skin rashes, except again for usually Q fever. Respond again to mostly of the tetracyclines. Most rickettsial diseases are zoonotic, meaning that they are animal-borne, so the organism that vectors it transmits it from an animal to the human, except for epidemic typhus, which is only found in humans. And transmission of these diseases is typically enhanced by poor hygiene, overcrowding of the animals and or humans, certainly wars, poverty, anything that would cause an increase in the vector population. This is the classic rash that we see with the spotted diseases like Rocky Mountain spotted fever and Ehrlichia diseases. And some of the ticks that can transmit Rocky Mountain spotted fever is the Dermacentor variabilis. This is the American dog tick that we have here in southwest Missouri. The Amebolomium americanum, the Lone Star tick, recognized by that little spot on the back of the female. The Rhizophilus sanguineus, the brown dog tick, which is very common here in southwest Missouri. Amebolomium maculatum, which is the Gulf Coast tick. This one is not quite here yet in Missouri, I don't think, but it's on its way because it's basically through central Arkansas at this point. And then lastly, Ixodes scapularis, the black-legged tick. This also transmits Borrelia burgdorferi, but can transmit both Ehrlichia and rickettsial diseases. This was reported rate of Rocky Mountain or of spotted rickettsial fevers from 2000 to 2013, and you'll notice that we have quite a number here in our region. And part of this is because, again, of the tick vectors that are here in this area. We have kind of another area concentrated over here in Virginia and North Carolina. We see a lot of Ehrlichia here. We see a lot of Ehrlichia and Rocky Mountain spotted fevers here in this area as well. Now, mycoplasms. These are unique in the fact that these are the smallest of all the bacteria. They are free-living, and they are the only wall-less bacteria. Primarily, they don't have a cell wall because they're just simply so small. They're about the size of a virus, basically about 0.3 to 0.5 microns in diameter. They do have a cell wall that is reinforced with cholesterols. The medical implications is because they have no cell wall, they stain very poorly. Penicillins and cephalosporins are not effective against these at all. And they actually reproduce outside living cells and can grow on artificial media. Mycoplasma pneumoniae is the primary mycoplasma bacteria we see, and this causes an atypical pneumonia, also sometimes referred to as walking pneumonia. This is most commonly seen in the winter. It's seen in younger adults, and oftentimes we see outbreaks within cloistered groups like in, college dormitories, other housing facilities. Typically see cough, sore throat, fever, headaches, malaise, just not feeling good, myalgia, muscle pain. You just don't feel very good. But typically the Mycoplasma pneumonia will resolve spontaneously in about 10 to 14 days. We'll respond to most of the tetracyclines still and to erythromycin. This is actually the organism itself, and you can see again, very small organisms. So it looks like they have a cell wall, but that's simply because they have a lot of cholesterol molecules in their cell membrane. When these are grown on artificial media, they actually tend to grow these spindle-like cells. And this is kind of the classic way we diagnose Mycoplasma pneumoniae, is by seeing these cells on the culture. Now, viruses. How do we classify viruses? Well, we can classify them as to whether or not they're carrying either DNA or RNA. And this is just the generalized structure of a virus. We will have a nucleic acid core, either DNA or RNA, that will be surrounded by a protein coat known as the capsid. And the capsid is composed of many single proteins called the capsomeres. Some viruses will then have a phospholipid envelope, which will have spikes, and these spikes will be involved in their attachment. Or some are just simply naked or non-enveloped, and the spikes will be on the surface of the capsid. The size and complexity of the viral genomes vary. Typically, the viral genomes are much smaller than bacteria. They are typically smaller than most other organisms, somewhere between 20 to about 300 nanometers in diameter or in length. The largest virus is actually Ebola, which is about 970 nanometers or almost 1 micron in length. They cannot be seen under the light microscope. And the nucleoid, or the nuclear core here, contains either DNA or RNA, but never both. So we can classify them as DNA-carrying viruses or RNA-carrying viruses. The capsid is simply that protective protein shell, and I really should say shell here, that surrounds the genetic material. The capsid can take on various geometrical shapes. They can form a helical structure, or they can form kind of this helical structure, or not helical, but isohedral structure around the protein, the nucleic. And here we see varying sizes of these viruses. So over here at the side, we have a eukaryotic yeast cell. It's about 7.5 microns in diameter. Streptococcus, about 1 micron, or 1 micrometer in diameter. E. coli, about 2 to 3 microns long, about 1 micron across. So this T2 bacteriophage, about 65 nanometers. Here we have the size of the hemoglobin protein, about 15 nanometers, and you can see that one of the flaviviruses that causes yellow fever is at about 22 nanometers in size. Polio is about 30 nanometers. The adenovirus is about 75 nanometers. Influenza about 100 nanometers. The current coronaviruses actually are what we call positive sense RNA carrying viruses, and they range at about 125 nanometers in diameter. Viruses are obligate intracellular parasites. Now, are viruses truly living organisms? Well, by the true definition of what a living organism is, viruses are not. Okay. They do not reproduce independently of a host cell. They don't do their own metabolism. They don't do anything else that a living organism cells do. And I'll make this statement. All living organisms, as far as we know on planet Earth, contain DNA. So the RNA carrying viruses, are they living? Well, again, not by the true definition. So I will refer to viruses as active, able to reproduce, or inactive. They have to reproduce or replicate with inside a living host cell. They cannot replicate outside of their host cell. Viruses lack metabolic enzymes, so they rely on the host cell's metabolic processes for survival and reproduction. They do not have a nucleus. They only contain DNA or RNA. They don't have ribosomes, mitochondria, or any other of the cellular organelles, and they cannot synthesize their own proteins or generate energy. They do not multiply by binary fission or mitosis. So therefore, they are truly not living organisms in my book. However, some people will say, but because they do reproduce, well, they can be living organisms. So as you listen to all of the wonderful fuss about coronavirus, they'll talk about, oh, the virus is living. I tend to disagree just a little bit with that one. So how do viruses replicate in animal cells? Well, first of all viruses, in order to replicate, have to first attach to their cells. They typically will do this using the spikes on the envelope or the capsid and attach to receptors on the surface of their host cell. This is typically what makes these viruses very host cell specific, is because they'll only attach to the receptor on a specific cell. If that cell does not have the receptor, the virus cannot attach. Then we actually will penetrate. And in animal cells, oftentimes the binding of the spike to the receptor will cause an induced endocytosis. We then have to uncoat. We basically have to break the capsid shell open in order to release the viral genetic code. Due to the various forms of nucleic acids that these viruses carry, they will replicate the DNA or RNA to a form that then can be utilized by the cell's protein synthesis mechanisms for the translation and transcription of the viral genome into viral proteins. We'll copy the viral code, and then we can assemble the new virons, the infective virus particles. The cells will then be released from their host cell either by lysing the host cell, so this cell will just fill up with viral particles and rupture, or in some cases these viruses may actually bud. They will simply then undergo a process in which they will push along with the cell membrane and take part of the cell membrane with them. What this will actually do is then form that envelope. So what we'll see is that when the virus comes over here, it will push this membrane outward, and we will form basically a bubble here with the viral code. And then eventually what this will do is become an enveloped virus out here. Sorry for my drawing, it's kind of crude. How do viruses then act? Well, they're going to invade their susceptible host cell. And in some cases, they then become a latent viral infection, and they are asymptomatic. In other words, the viral code is simply in the host cell, but is not currently being transcribed into viral protein. So we're not reproducing the protein right at the moment. Or it could immediately cause acute cell necrosis and degeneration by rapid replication of the virus. It might cause cell hyperplasia and proliferation because we gear up to make more and more viral proteins. Or it could have a slowly progressive cell injury. This is something that HIV will do, is that first of all, HIV typically tends to be asymptomatic in the early stages. So while the viral code is in the host cell, it is not being made into new viruses. But as it does, or as we start to read the viral code and translate those viral proteins, then we get a slow progressive cell injury because HIV is released from the host cell by budding. And we also know that some viruses will actually change the genetic code of the host cell and result in a dysplastic cell causing neoplasia or cancer. Some viruses will form inclusion bodies within their host cells, and literally these are just groups of the viruses within the cytoplasm. So for example, rabies virus, 100% fatal if not treated. The only way that we make a definitive diagnosis of rabies is on autopsy when we look for the inclusion bodies of the rabies virus in the neurons of the brain. Now, how does the body defend against viral infections? Well, there's several different ways in which the body tries to destroy these viruses once they enter the body. One is we can form interferon. This is a broad-spectrum antiviral agent produced by virally infected cells to prevent infection of other potential host cells. And I'll talk about interferon here in just a moment. We can see cell-mediated immunity against virally infected cells using the cytotoxic T cells, and we actually can make antibodies against certain viruses. One of the reasons that HIV is so hard to make antibodies against is because we simply don't get to see the virus long enough to activate B lymphocytes and form antibodies. Because what we see is from the time of entrance of HIV into the host or into the body and into its preferred host cell, the T helper lymphocyte, is approximately 10 hours. Remember, it takes 7 to 10 days to produce that antibody response in a primary immune response. There are a number of antiviral agents out there on the market. Some of these will block viral multiplication within the host cell. Others will actually prevent the virus from invading the host cell, and these are known as entrance inhibitors or blocking antibodies. But the problem is most of these have limited application because of the toxicity to the host cell and limited effectiveness because many of these viruses will find ways around these drugs. Now, said we'd come back to interferon. How does interferons actually work? Well, here we have a virally infected cell and it's busy making viral proteins. But what we'll see in certain cells is the presence of the viral genome will activate a group of genes called the interferon-producing genes. These will produce interferons that are released out of this virally infected cell. They will go over to another susceptible host cell and bind to receptors. Now, two things can happen here. One is this interferon receptor binding signals to the nucleus where we activate a group of proteins called the antiviral protein genes. These will produce antiviral proteins that if the virus should come in, they will be destroyed immediately and will not be able to reproduce. The other thing is that the binding of the interferon to this receptor may actually be the receptor that the virus needs to get into the cell, and therefore interferon blocks the binding of the virus, so we do not get attachment. Right, what about the fungi? Well, these are plant-like organisms that do not contain chlorophyll, so they have to get their food from the surrounding environment. If they get their food from dead organic matter, they're known as saprophytes. If they get their nutrients from living tissues that they're on, then they are known as parasitic fungi. There are two forms of fungi: yeasts, which are the single-celled fungi, and molds, which are the multicellular fungi. Most are obligate aerobes, and they typically tend to be opportunistic organisms. The natural habitat typically is in the environment, except for the Candida species, which do tend to only grow in or on living tissue. The cell wall of fungi is made up of a protein called chitin. This is the same protein that makes up the hard exoskeleton of shellfish versus the peptidoglycan that we find in bacteria. And the cell membrane actually has a number of sterols, but it's not cholesterol. It's known as ergosterol and zygosterol. These are simply other sterol molecules that are in the cell membrane of the fungi. So here's our little yeasty beasties, and these are actually busy reproducing, producing what we call buds. And this right here is what's known as the bud scar, where a previous bud has broken off. This is a common mold, bread mold. And yes, mushrooms are actually the fruiting or reproducing body of a mold. The mold is actually in the ground or in whatever material it's getting its nutrient from, and the mushrooms are actually the reproductive bodies that we see above ground. Most fungi like high humidity, so moisture. They like a little bit of heat, and they typically like dark areas with a good oxygen supply. There are a number of antifungal drugs that are available, and so some of these fungi are actually beneficial to us. So where would we be without the fungi that produced beer, cheese, wine, and beer? Frequently, fungi will be associated with decaying organic matter, and oftentimes molds we find involved in the spoilage of food, so like your bread molds, or fruits, or grains, or other vegetables. I suspect right now there's a fungus that's growing in my refrigerator somewhere in the back. Infections on living tissue we can actually classify as to the level of the infection. So the superficial fungal infections will infect the hair, the skin, the nails, um, the mucous membranes, okay, and the cutaneous infections get down a little bit deeper. And then we have the dermatophytes that get down into the level of the dermis. And then we have what's called the systemic fungi. All four of these will be inhaled. And so I'm going to talk a little bit about histoplasmosis, coccidiomycosis, blastomycosis, and cryptococcosis. Coccidiomycosis is caused by the organism Coccidioides immitis. Coccidioides immitis, and these are the actual yeasts of this organism in lung tissue, will be inhaled from soil. This can result in a severe pneumonia-like disease within the lungs. And many of these, in fact, all of these systemic fungal infections tends to form what we call a snowstorm pattern in the lung field. So it kind of looks like it's been snowing there. Coccidioides immitis is found in dry desert alkaline soils. So we primarily see coccidiomycosis out in the southwestern United States. This can actually show a Coccidioides immitis lesion here on the tongue, and this is actually the organism here. So this is causing an ulcerative lesion on the tongue. This is sometimes known as valley fever or Rift Valley fever. And this is the distribution map for endemic areas with coccidioidomycosis within the United States. So again, out here in the dry arid desert regions of New Mexico, Arizona, Southern California, Southern Nevada, and Utah, and in northern Mexico. Histoplasmosis caused by Histoplasma capsulatum, and this is again the organism. It actually at room temperature forms kind of hyphae-like structures of a mold, but in the lungs or other tissues it will form yeast. Also again causes a pneumonia, respiratory-like disease within the lungs, but can go systemic where it can settle in joints. It can cause skin lesions similar to this on the legs or other parts of the body, and can actually result in kidney damage or liver damage. Blastomycosis caused by Blastomyces dermatitis is also a yeast that we find in the lung tissue here, and again causing that fine snowstorm pattern of lesions within the lungs. But if it does go systemic, oftentimes we can see skin lesions with blastomycosis. And this is kind of a common lesion that we would see, oftentimes associated around the oral and nasal passages. Both histoplasmosis and blastomycosis have overlapping areas that we see uh, for their distribution. For histoplasmosis, the Ohio and Mississippi River valleys, and so we do see endemic areas here in southeastern Missouri, but also we see this here in southwestern Missouri. Histoplasmosis is most associated with breathing in dried bird feces and dried bat guano. And so some of our cavers in this area oftentimes develop histoplasmosis. Blastomycosis also Ohio, Mississippi River valleys, but does extend a little further north than histoplasmosis. Blastomycosis is most associated with breathing in dried bird feces, and so concentrated poultry production houses are a notorious spot for blastomycosis transmission from the bird feces to the workers within the house. A few years ago down here in northern Arkansas, we actually had several workers at one of the Tyson poultry plants that came down with blastomycosis, and so they had to depopulate the birds within those houses and actually burn the houses to the ground to get rid of the blastomyces within the soil and in the bird feces. And then the last of these systemic fungal diseases is cryptococcosis, caused by Cryptococcus neoformans. Cryptococcus has a worldwide distribution, and this is a classic way we diagnose Cryptococcus. This is the actual yeast, and Cryptococcus then has this very large protein capsule around it. So we do a negative stain on a slide, and we see this big thick capsule around this yeast. We know that we have Cryptococcus neoformans. Again, in the lungs, causes that kind of snowstorm pattern that's going on. This is a highly consolidated lung with pneumonia here caused by Cryptococcus. While the other three are actually more rural diseases, Cryptococcus tends to be more of an urban disease. And the reason for this is Cryptococcus neoformans is most associated with breathing in the feces of this little guy, the pigeon. And so think about where these are concentrated. They tend to be more in urban areas, so they sit over the doorways of buildings and they, do their business. That feces dries, you walk across it, aerosolize it, and breathe it in. So while Cryptococcus is found throughout the world and it's more of an urban disease, the other three, histoplasmosis, blastomycosis, and and not cryptococcosis, but cryptococcidiomycosis is actually more rural. Thank you.